You're listening to the Skeptic's Guide to the Universe, your escape to reality.
Hello and welcome to the Skeptic's Guide to the Universe. Today is Thursday, October 2nd, 2025, and this is your host, Stephen Novella. Joining me this week are Bob Novella.
Hey, everybody.
Kara Santamaria.
Howdy.
Jay Novella.
Hey, guys.
And we have a special guest, Lee Mossbacher. Lee, welcome to the Skeptic's Guide.
Thank you for having me.
And Lee, you're joining us because you are a patron of the show, and you just mentioned to us that you've been listening to us for 20 years. Is that really true?
Well, when did you start? I think it was like 2005 or 2008 or
2005. So. We started in 2005.
Well, I was part of the forums for the longest time and a listener on my iPod, then my iPhone. So, yeah.
Okay, cool. Because we only had like 200 listeners back in 2005.
Well, I did win one of the Who's That Noisy, I think.
Oh, we could go back into the email archives. Okay. That's amazing.
Yeah, to make it clear, just so people that are listening know, so you have to be a patron at the $200 level or higher in order to get that. That's why we don't have that many people who actually get to do this. So first off, Lee, I just want to thank you for being an awesome supporter of the SGU. During these dark times, we need support because, we are trying to expand our reach and we're starting new podcasts and, adding in more live stream content, all to in some way help chip a little bit away at the amount of insanity that's going on.
So you may notice that Evan's not here this week. That's mainly because his internet is down. We tried to troubleshoot, but he had other things going on too, so it just wasn't going to work this week. But that's okay because we got Lee to take his place.
Oh yeah, it's Yom Kippur is just starting, so
yeah,
I don't know if you're supposed to say happy. Yeah, I don't know if Yom Kippur is a happy holiday, but
aren't all Jewish holidays not happy holidays?
No, I think that Rosh Hashanah is pretty happy.
Okay.
So, but I never know which ones to say, like, happy this, or if it's like, and a good Yom Kippur to you. I'm not sure.
We need to talk about Jane Goodall.
I cannot, man. Well, I mean, I can believe it. She was, 91, 92?
91.
Yeah, that's...
Oh, boy.
Yeah, she died yesterday, October 1st. So most people here know who Jane Goodall was. She was essentially famous for being a primatologist, and she single-handedly transformed our understanding of chimpanzees with an over, I think, six decades of field research living with chimpanzees. Again, really changing our understanding of how human, really, chimpanzees are, how sophisticated their behavior is, their social structures, etc. And she also became a very, very spoken advocate for wildlife, for animal rights, also for mitigating climate change, etc. Very active right up until the end.
You know something really interesting about her that I didn't realize? So, I think about Jane Goodall and Diane Fossey and like these women naturalist scientists who spent all this time in the field doing incredible work. And I think famously Diane Fossey was a scientist for sure, but she wasn't a scientist in the traditional sense, as she wasn't like an academic scientist. Jane Goodall, she earned her PhD from Cambridge in 1966. She was admitted to her PhD program in 1962, but did not have an undergrad degree. But they accepted her because she had done so much important field work already with chimps. So this was like an era of women having a hard time, right, getting into these programs or getting any power within these programs and just saying, screw it, I'm just going to go do the work.
Right.
Which is incredible.
And, to his credit, she had a lot of support from Louis Leakey. Basically, I think you got her started on her.
Yeah, I mean, and that's project. Very often what had to happen.
There. Yeah.
Yeah, women had to find doors, find someone, find help to open that door and push through it and never look back. And she, obviously, her body of work is incredible.
Yeah, I agree. Absolutely incredible. So I remember I was reading a science fiction book about uplifting chimpanzees into sentience.
A la Brin, David Brin.
I think it might have been Brin. And the chimpanzees, like their expletive was Goodall. Like that's what they would say.
Very cool.
Oh my God, I love it. I forgot about that.
You remember that? Yeah, that's funny.
I wonder if that book holds up.
Nice touch.
Yeah.
Okay. So, Bob, you're going to start us off with a quickie.
Thank you, Steve. This is your Quickie with Bob. I love finding quirks to the shape of cosmological objects that we used to think were relatively simple, right? The iconic example is the Earth. It's a sphere, right? But no, it's not just a sphere. It's an oblate spheroid, like if you squished it so that the equator bulges out a bit, right? But then beyond that, an even more accurate shape to the Earth might be described as a lumpy geoid due to those, all these irregularities to the shape caused by the gravitational field differences, oceans, land masses, all that stuff. So our galaxy is similar. For years growing up, it was the Milky Way is a spiral, right? But then it became a barred spiral with a rotating bar going through the center. And the classic spirals of our galaxy emerged then from that bar. Also, if you could see gamma rays and X-rays, which would be quite cool, I think, you would see, and you were looking at the Milky Way, you would see massive lobes of radiation billowing above and below the core of the galaxy. So now I'll be talking about another twist, a galaxy-wide wave rippling through our galaxy.
Bob, can I, before you go on to that?
Sure.
You could also say that our galaxy is twisted. It's not perfectly flat.
Yep, it's definitely not a pizza shape as I came across in some of this research. So yeah, there's even more little nuances and quirks to the shape of our galaxy. But the biggest new reveal about the shape of the galaxy now is this galaxy-wide wave rippling through it. Billions of stars just bobbing essentially up and down like a galaxy-sized stadium, right, of a crowd doing the wave. It's kind of like what's happening at a huge scale in the Milky Way. This was discovered from the Gaia Space Telescope. Gaia mapped the motions of millions of stars to reveal this pattern. And it did this before it pulled a Steve and retired earlier this year. So it's a last interesting discovery by the Gaia Space Telescope. The reach of this phenomenon extends from 30,000 light years to 65,000 light years from our galactic center. So it's extremely widespread. And we're not sure what causes this wave, but there are models, and those models point to the Sagittarius dwarf galaxy, which may have passed through the Milky Way multiple times, hundreds of millions of years ago, creating these ripples that we're detecting now, kind of like a huge rock being repeatedly dropped into a galaxy-wide pond. So fascinating stuff. Look it up online. This has been your Rippling Quickie with Bob. Back to you, Steve.
All right, thanks, Bob. Lee, before we go on with the regular news items, tell us a little bit about yourself.
For sure. So I guess my I'm from Ohio. I grew up on a farm, and I would say that, my early life was tumultuous, and I would say that physics and Star Trek saved my life.
Mm-hmm.
And so I got really interested in physics and applied to university, got accepted, did 4 years at a local university, took a break to start a company, sold that company, and then went back to get a PhD.
Nice.
Wow.
And from that, got into investing and running a hedge fund, doing some startup investments. And that's what I've been working on the last 20 years.
Oh, wow.
What was your PhD in specifically?
It was more material science. It was physics, but I studied a material system called zinc oxide, and was the first to create a P-type conductivity in that material.
Okay.
Yeah. Mm-hmm. Had a lot of applications to like solar detectors, ultraviolet lasers, and things like that.
And tell us how the SGU contributed to saving your life. Just assuming that it did. But
you joke about that, but I would say that the SGU taught me more about scientific philosophy and critical thinking than 8 years in grad school.
Wow.
So is that a good thing about us or a bad thing about American educational system?
Definitely the American education system.
Yeah. I mean, obviously, we talk about this all the time, where it doesn't necessarily teach you critical thinking. I know brilliant scientists who have these glaring blind spots in their metacognition because it's not systematically taught. You know what I mean? Which is a real lack in our education system.
Absolutely, it totally depends on which program you're in, which professors
your
you
mentor.
have.
It's really mentor dependent.
Right?
Totally. Some people really do get a great education in that area, and some people do not. It bums me out that very often most science degrees in the U.S., not all, doctoral degrees are PhDs, which is a doctor of philosophy, and very few people actually get any philosophy training.
Mm-hmm. Got zero philosophy training. All of it was in the SGU.
Wow.
I don't think I heard Popper's name until 2005 or 2006.
Wow.
Yeah, you would think the philosophy of science would be one of those 101 things you would learn when you start to study science. But you learn biology, chemistry, physics, but not not how to do science or the philosophy of science. They teach it like in 5th grade, but so poorly that it doesn't really have any effect, in my opinion, on the general public's understanding of science.
I took a wonderful philosophy of natural science class in my training, but that's because I was a psychology major and I was a philosophy minor. I think the philosophy of natural science class was offered in the philosophy department and didn't offer any science credit. And then my philosophy of psychology in the mind class was dual credit for philosophy and psychology. Like, come on, natural sciences.
Yeah.
Let your students take this class, even if it's in the philosophy department, and give them credit towards their degree.
Yeah, I agree. All right. I'm going to talk to you guys about redrawing the family tree of humans, which is something that happens quite frequently because you guys have heard the muddle in the middle. Have you ever heard that term?
No, what is that?
Yeah.
So we know a pretty high resolution picture of very recent human evolution. And we've really pieced together a lot of sort of the earlier hominid evolution. But there's this zone about 1 million years ago where we still don't have a lot of specimens, and there's a lot was happening too. There's a combination of seems to have been a period of rapid diversification for which we have few fossils, and so there's just a lot we don't know about it, right? And so, but a few years ago, these 2 fossils were discovered in China, the Yunxian 1 and Yunxian 2 fossils. These are humans, right? They're Homo whatever, and they're fairly complete, but they were in not great condition because most, fossils are crushed, right? I mean, we don't find them in perfect shape in the ground. We find them in compressed fragments.
Yeah, dirt and rock weigh a lot,
They weigh a lot,
and millions of years.
Been there for a long time. So one thing I didn't, if I knew this, I forgot it, but what I didn't know explicitly was that there's different types of distortion that can happen to fossils.
Cool.
Yunxian 1 fossil had what's called plastic distortion, meaning that pressure over a very long period of time has actually changed the shape of the bone, which is bad, right? That's the bad, that's like the worst kind of distortion. Like you could imagine, like it didn't just crush a skull fragment, it actually flattened it out because it changed over time due to the pressure on it. The Yunxian 2 skull didn't have any of that plastic distortion. It had a lot of fracturing and then displacement of the fragments. That's an easier type of distortion to fix because theoretically you could just reassemble the fragments.
Yeah, big 3D puzzle.
Yes. Now the new news item is, again, these skulls, Yunxian 1 and 2 were found years ago, but paleontologists have used new technology to examine them, and especially the 2 skull because that's, again, in better shape. Although they did use some information from Yunxian 1 to fill in a couple of gaps here and there, but most of the information came from 2. And it's essentially using CT scanning and a specific technique called CT segmentation and fragment realignment.
Ooh.
So they use a CT scan to take highly detailed X-rays of the fossil, in its natural state, and then identify the fragments and then digitally realign them.
Little AI involved there?
I would imagine so, but it didn't explicitly say that. But at least not that I saw. So that allowed for a much better reconstruction than we have been able to do in the past. In the past, one thing you have to understand is like when we were just physically trying to piece together fossils. And again, there may be some plastic distortion. There's a lot of fracturing. There's crushing. There's a lot of guesswork involved, more than you might think. You can make those pieces fit together in different ways. But unfortunately, when you're talking about, again, like this phase of human evolution, whether or not that skull is one species or another species depends upon subtle anatomy of the skull. And so just doing a regular reconstruction, paleontologists were unable to answer the question, which clade should we put this skull into? Because it all depends on how you put those pieces together, and it's a judgment call. There was no objective way to answer that question. But with this technique of a CT segmentation and fragment realignment, they were able to do such a high-quality reconstruction that they could answer that question, that they could say these subtle anatomical features place the skull into this clade rather than that clade.
Nice.
Having said that, what does it tell us? So the skull, Yunxian 1 and 2, right, with this reconstruction, they believe that they fit into the clade Homo longi, Longi, which is a sister clade of Homo sapiens. So the way the branching order goes is that there was a common ancestor of all of the modern Homo clades, basically Neanderthal, Longi, and human. That split off into Neanderthals in one branch, and then Sapiens and Longi in the other branch, and then they quickly branched off from each other. And this goes back 1.2 to 1.1 million years ago when those two branchings occurred.
So that's kind of like the common ancestor for gorillas, chimps, and humans, where the gorillas branched off first from the common ancestor, and then chimps and humans, Homo sapiens, hung out together for a while, and then they branched.
Same relationship. So 1.2-ish million years ago, Neanderthals split off. 1.1 to 1 million years ago, Longi and Sapiens split off from each other. And then the Denisovans, you guys know the Denisovans?
Yeah.
They're probably in the Longi clade, the Homo longi. And Homo longi is basically Asian humans. That's they existed in Asia, which is why these skulls were found in China, right?
Okay, what was the common ancestor, though, between...
Well, we don't know. That's the muddle in the middle, that we don't have that common ancestor of all three.
That's huge, man. How the hell do we don't have that?
But that happens so much in evolution.
But this is important. This is us.
But yeah, again, a lot's happening. Clearly, very quickly. There's probably so many sub branches that we haven't found so far, and it's happening very quickly, and also in isolated small populations, almost by definition, right?
Yeah.
So that what this means. So I didn't state a very important fact here. How old is this skull? How old do you think this skull is?
1 billion years old.
No, of course not.
That's a little bit too much.
400?
It's about 1 million years old.
Oh, wow.
It's just about 1 million years old. So it's pretty much right at the branching point of Homo sapiens and Homo longi. And they place it in the Homo longi branch. And in fact, this skull is establishing when that branch took place.
That was a long time ago.
That was a long time ago.
Yeah.
So what this means is that the Homo sapiens clade is about twice as old as we thought it was. The prior dating was that last split into Homo sapiens, into a clade we would call Homo sapiens, took place about 500,000 years ago. This new classification of the skull suggests that, nope, the Homo sapiens clade branched off 1 million years ago.
That's nuts.
That is nuts.
What the hell?
Yeah.
Now,
if we're that much older, what the hell took us so long to get where we're at?
That's a good question.
But, million years.
But hang on, these are still archaic humans we're talking about. Even though they're technically Homo sapiens, they look different than we do, right? They are.
Yeah,
they are.
I think we would think they were
humans.
apes.
No,
if we saw them in the wild.
No. If you saw,
I think so. Even when I see Lucy, it looks like a very human-like ape.
Well, we are so far away from Lucy.
Yeah, that's true. Because you're talking about that split.
The cliche is that the conventional wisdom among paleontologists is if you saw a Neanderthal in the New York subway station dressed in modern clothes, you would not blink twice. Yeah, you would not necessarily notice them. So with these, like with Homo sapiens and Homo longi, maybe their eyebrows were a little bit generous, their forehead sloped back a little bit more and they had maybe a little bit more elongated occipital thing. That's what you would notice. And there's a lot of little real subtleties that only a paleontologist would
Could
notice.
they have a sagittal crest?
No, nothing like. No, that's only in the robustus branch of the Australopithecines. That's like that's millions of years more separated.
That anchored the powerful chewing muscles.
The chewing
right?
muscles, yeah. That's only the robust Australopithecines, basically.
It looks like Australopithecines is 3 to 2 million. But this is nuts that this is all already. I guess that's why I was thinking already 1 million years ago. How human did they really look?
Yeah. Again, that's what this is telling us, that this was 1 million year old skull was very close to the branch point of the Homo sapiens clade.
I guess, though, we were probably still covered in hair at that point, right?
No, probably not.
Oh, you don't think so? Okay.
No. Huh. Once we started living in groups, wearing furs and stuff like that, there would have been selective pressure
against it.
Yeah, against that. We don't know for sure, but I'm not hairy like apes are hairy, not like chimpanzees are hairy. But that's one of those things that it's hard to know for sure.
Mm-hmm. Don't we have more hair follicles than gorillas?
I think it's the same. It's just how big and thick the hair is.
Yeah. Okay.
So interesting. Yeah, no, I've studied human evolution in college. I've been following it very, very closely, constantly redrawing the tree in my head with new information coming in. And this is a big change. This is a pretty big change.
It's getting very complicated. No DNA made it, right?
No, not 1 million. I think 1 million is too much. It's like we have Neanderthal DNA, but it's from like 40, 50,000 years ago.
Are any of these stuck in amber?
Yeah. It wouldn't help. That doesn't stop the DNA decay clock. It has a half-life.
Are any of these stuck in the Jurassic Park movie?
So interesting, and this is not the end, obviously, and even the implications of this fossil, will continue to be debated, because again, it's really coming down to some subtleties of anatomy and exactly how the reconstruction was made. So this is the best reconstruction we have so far because we're using new technology. By the way, I have multiple news items queued up about fossils where this technique is sort of making huge impact on our ability to, So, and one thing though, like I also was looking at a news item about, again, another specimen that's at the beginning, the branch point of snakes and lizards. So pretty much the same story, but for snakes and lizards. And again, it took 10 years. It took 10 years to do this analysis of that fossil. This
10
CT.
Years?
10 years. So this is a very detailed kind of reconstruction that we can do with the CT scanning. But it takes time. But the results are worth it, because it's really transforming our understanding of some very key points in evolution. All right, let's move on. Jay, are there wasps in my figs?
All right, so before I get to it, I got to ask you guys a question.
Yeah.
Well, I want to ask Lee a question. Lee, can you hear me?
Yes.
Okay.
That's the question.
And you're sitting down, correct?
I am.
All right. Now, be honest. Have you ever eaten a wasp?
No, I don't think I have.
How sure could you possibly be?
I would say 30% sure.
I would like to hear from everybody. Have you ever heard of someone saying that figs contain dead wasps?
No.
Not until
is that like a...
No, not really.
Seriously?
Is that
Bob?
like a common...
How about you, Bob?
I never heard of that.
Now I'm terrified.
Okay.
I mean, but to be fair, eating figs is rare in my experience. Usually it's like fig Newtons, but just regular figs, it's not something that I eat often. And
what about figgy pudding?
No.
Well, this is a news item.
Jam?
This I chose this news item, guys, because a lot of people out there have heard this urban legend, like it's something that people just say, that if you're eating a fig, in order for the fig to exist, a wasp had to crawl inside the fig.
Oh,
what?
Geez.
And I literally am shocked that none of you have heard this. Like I've known this for a really long time, because I was going to ask you whether or not you think it's true. So would you guys believe this if somebody said to you, hey, did you know that fig you're eating, like a wasp had to crawl in there for the fig to exist? Do you think that's true or false?
I don't think I would buy that.
Yeah, I'm leaning towards false. I mean, unless I hear the reason, what reason could there be requiring the death, the suicide of a wasp for a fig to even exist? What does it need? Is it some sort of
there's an answer.
parasite scenario? What's going on?
I'm glad in one way because I'm going to tell you guys all about something that you know, first off, you didn't know about the urban legend. And second of all, you don't know any of this. And it's really cool. And it's a really great example of how complicated nature actually is. So first, let me explain to you what a fig is, right? So you guys would think, what? It's a fruit, right? So botanically speaking, it is a fruit, but it's in the usual sense, you could say that a fig is called a synconium. This is a hollow, fleshy structure packed with hundreds of tiny flowers on the inside. I bet you didn't know that about figs.
No.
So what looks like, smooth, self-contained, fruit flesh that's inside, it's really an enclosed floral chamber, which I think is just fascinating. Like, when you open up a fig, you're like, there's the fruit and it's sweet, but it isn't simply just that. The design here sets the stage for essentially one of the most interesting and peculiar relationships in the natural world, with no exaggeration. So now we've got the fig wasp. So for tens of millions of years, maybe old enough for the creatures that Steve was describing to live during when this was first coming to be. Figs and fig wasps, they co-evolved and they have a really tight relationship. So about 850 fig species exist, and almost every single one of them has a matching wasp species specifically that serves as its pollinator. So the wasp, they depend on the figs for reproduction, and the fig depends on the wasps to carry pollen from one synconium to another, right? Essentially the same way as other flowers need this same activity. Like bees do it, other insects do it. There's lots of different ways that it's carried out. But in the fig world, a wasp really is the only thing that can do it.
Birds do it, bees do it. No, okay.
Kara, what is this called, by the way? Does anybody know what this is called?
Synergy?
It's called biological mutualism.
Mutualism, yeah. Yes.
Yep.
And why isn't it just symbiosis?
Because they use more complicated terms to make it sound important. This one, though, this particular biological mutualism has a really wacky twist, which is the fun part. So when a female wasp finds a receptive fig, the wasp will crawl inside through a small opening called the ostiole, and it's almost as if these two things evolved to interact with each other. That little hole and that tiny little wasp, I don't know, Steve. God of the gaps, whatever, man, it's happening. So one of two things can happen. If she enters a male fig, which is called a caprifig, she lays her eggs in some of the flowers that are inside, and then her offspring hatch. They mate. And then they eventually, get out and they create a new generation of the females that fly off, and they repeat the cycle. If she enters a female fig, which are the edible kind, this is an important delineation here, things don't go as well for the wasp in one sense. So she pollinates the internal flowers. Like she's crawling through the hole. Her wings and her antennae usually get stripped off. She gets in deep enough to pollinate the internal flowers, but she can't lay her eggs because the flower styles in the female fig are much longer than the ones in the male fig. So the female wasp has something called an ovipositor, and this is a slender tube-like organ. You might have seen some insects with these. They use them to insert eggs into the ovules of the flowers. And that's essentially like where it would put the eggs in order for it to grow if it went into a male fig. But because the size of the flowers inside the female fig are really long, she can't reach the right spot. So what happens is she dies inside, and she leaves behind, of course, her body, but no eggs or anything like that. Okay, so what happens next? Because here we are with the edible figs, the female figs, with a dead wasp on the inside.
Somebody eats the wasp.
So is that your guess, Steve?
Well, they're nice and tangy, I hear.
Yeah, they have a little bit of tang to them. Anybody else have any guesses of what happens next here?
To the wasp?
To everything, just this whole situation. You have the dead female wasp in a in a living edible female fig.
Yeah.
The fig probably processes the protein somehow.
Kara, I love you in so many ways.
It's probably, it's carnivorous until it isn't, until we eat it.
Everybody take note of how smart Kara is. So the question is, if the wasp dies inside the fig, does this mean that people are actually eating insect corpses every time they bite into one? And I have to tell you, I've eaten some straight up female figs in the last few years of my life, and I have had this bouncing around in my head like, all right, I'm sure I eat lots of insects, like all the time. You know, it's hard to avoid in the modern world.
Yeah, but I don't give a fig.
Gotta fill in that Evan.
Gotta fill in for Evan,
distance. Yeah, exactly.
All right, but this even is more complicated because if you're asking the question, do people eat wasp corpses inside of the figs that they're eating? The answer depends on the kind of fig. So here's another layer of complexity. Most of the figs you see, for example, in American supermarkets, the Mission fig, the Cadota, the brown turkey, these are all parthenocarpic, which means they don't require pollination at all. These figs ripen and develop without any wasp ever entering into a tiny hole and stripping parts of its body off and dying, right? So the majority of the figs people eat never had a wasp in them in the first place. When I read this, I was very relieved. Not that it would change my life in any way, but it just made me feel good that I'm not eating wasps of any kind.
But I pretty much only eat medjool. No, those are dates. Oh, yeah, I don't know what kinds of figs I eat. When do people eat figs?
I grew figs on my deck. I grew them for several years, but they died over last winter. I don't know why that happened. But that type of fig tree does not require pollination.
Okay, so you had more of more of the safe ones to eat. Okay, so
not that the other ones are unsafe. I mean, that's not what we're implying here. But it didn't require a little wasp to crawl inside and die.
So, of course, there is an exception to that list because the Kelly-Myrna figs do require pollination by a fig wasp. And in those cases, it's possible that a female wasp does die inside. But there's even more detail here. And this is the part that Kara had some insight into. Figs produce a milky latex that has proteolytic, you ever hear of that? Proteolytic enzymes? Mm-hmm. The one that we're talking about in particular is called physin. And these enzymes break down proteins. And they don't really care about, whether it's plant tissue or insect tissue. They just break down proteins. So when people have studied this, they look at the fig latex and they confirm that the physin, the related enzymes, they are indeed powerful enough to quote unquote, digest the soft animal matter that's inside the fig. So even if a wasp does die inside, its body just doesn't remain intact. It gets, by the time the fig fully ripens, what's left it's been broken down into amino acids and it's been absorbed into the surrounding flesh of the fruit.
Yeah, but those are wasp amino acids.
But still, I mean, there's a big difference between having an amino acid in your mouth and a frickin', insect leg.
Yeah, they all get turned into amino acids eventually.
Yeah, whether it's in your mouth or your stomach.
But you're not biting into a bug. And that is the clencher of this whole thing, Steve. That's why the people who have heard what I have heard wanted to hear this. You are not biting into a bug of any kind unless another one got in much later in the process, which is unlikely. So I personally feel happy and relieved for all the fig eaters out there that they're not eating bugs.
So that was the upshot of this whole news item is just don't worry, you're not eating bugs when you eat figs.
So even if you were eating bugs, you would be fine.
But I want to tell you how important figs are because outside of fig Newtons, maybe most people have no use for them, but figs are really, really important. The fig-wasp relationship is some weird, odd trivia fact, and it's good, it's cool, it just demonstrates how awesome nature is. But ecologists consider figs to be pretty much like a keystone species in the tropical forest because, figs can fruit year-round. And because of that, they sustain birds and bats and primates, particularly during periods when other food is largely not available. So without the figs, a lot of these ecosystems would just straight up collapse. And without wasps, there would apparently be no figs. So we need that relationship. The figs are important because it feeds a ton of animals, and you can't have fig Newtons without figs. You guys feel like you learned something?
Oh, sure.
Yeah.
Cool.
Great.
I was like 3% confident that fig Newtons actually had figs.
Yeah.
When I said that, Lee, I was thinking there might not be, but the one thing about fig Newtons that is you're definitely crunching down on seeds in there.
Yeah.
Because there is a little bit of that.
Yeah, but thanks to you, Jay, when I crunch down on that, I'm going to be thinking of wasp feet and wings and crap. Thank you.
Yeah, but empirically,
for the record, fig Newtons do contain figs.
Of course they do.
The filling is made of dried figs and fig paste.
For all of my elder millennials out there who watched Saved by the Bell growing up, you may remember the episode where Zach Morris freaks out his teacher because he serves him chocolate-covered, I think, crickets. And he crunches into it, and he's like, "Oh, it's so good and crunchy." And he goes, "The crunchy part's the thorax." And that has been drilled into my head since I was a child.
All right. Thank you, Jay. Kara, tell us about ALS and whether it may or may not be an autoimmune disease.
Well, I'm going to tell you what a recent Nature article told me, and then Steve's going to tell you why you should be skeptical, or maybe not skeptical, but take it with a deep breath and a grain of salt. So a recent article that was published in Nature, actually yesterday in Nature, titled Autoimmune Response to C9. I don't know if you're supposed to call this protein C9ORF72, or I think it rolls off the tongue more to say C9ORF72. Usually scientists do that, so we'll see. Autoimmune response to C9ORF72 protein in amyotrophic lateral sclerosis. Okay, so that's, I don't know. Most people, they come across that title and they go, okay, I don't know what that means. So let's dig into it. So what we do know about ALS, and actually we know a lot more than what I'm about to say, is that it is a neurodegenerative disease, and it's characterized by progressive loss of motor neurons. We also know that there are multiple mechanisms that appear to be at play, but the big picture is still a little bit fuzzy. So
one minor point of clarification. It's specifically, it's both upper and lower motor neurons, because there are different diseases that cause just lower motor neuron loss or just upper motor neuron loss. ALS is defined clinically as it has to have both.
As both. Okay. And for clarity, upper is brain to spinal cord, right? And lower is spinal cord to muscle. Yeah.
It's literally a two neuron system. There are two actual neurons. Yeah.
And so, after, years and years and years of research, we do know that there are some processes that are involved and that there are some disease mechanisms that have been identified, but we still don't know kind of the why of it all and how all these things fit together. We know that there are changes to the structure and the dynamics of the axons in ALS. We know that there are both apoptosis and necrosis, so programmed cell death and cell death due to damage or inflammation. We know that there are changes to the mitochondria in ALS. We also know that some neurotransmitters are affected, like glutamate. And we know that inflammation is a big part of the process. But according to the ALS Association's website, there's literally a sentence here that says, inflammation in the central nervous system, it's called neuroinflammation. There's increasing evidence that neuroinflammation accompanies the death of motor neurons in ALS. However, evidence so far does not support that ALS is an autoimmune disease. The inflammatory process apparently is a reaction to the death of the cells and not the instigator. But this new study in Nature is claiming that may not be the case. And so we'll talk about whether or not that is true soon. So basically, in this study, what the investigators found, they tried to kind of chunk out the ALS patients that they looked at into two different groups. The ones who have a shorter time to death from diagnosis and the ones who have a longer survival time from diagnosis. And they wanted to see, is there something different about these two groups? They specifically looked at that protein that I mentioned before, the C9ORF72 protein, because they knew that it had been implicated in ALS. And they found that in these patients who had a quicker time to death, their CD4 plus T cells were attacking the proteins, that specific protein I mentioned, on their neurons in large numbers. And they found that in the group that had a longer survival time, they had more anti-inflammatory T cells present. So they called that like a protective immune response. And so in their view, I mean, we can get deeper into, the what are they called? Like the epitomes, like the antigens on those cells that they were able to identify, how they attacked them, how they binded, all that good stuff, all of the inflammatory factors that kind of happen downstream as a result of that. But the big sort of takeaway here is that in their view, what's one of the main drivers, I mean, it really does feel like that's how this is positioned. One of the main drivers is an autoimmune response. So your body has these proteins on these neurons, right? These C9orf72 proteins, and you have these CD4 plus type T cells. In ALS, they're claiming that they are actively attacking those proteins on the neurons, and that in a small subgroup of ALS patients, they have anti-inflammatory T cells that they produce in response to that gives them a protective immune response that helps them live longer. That's sort of the long and short of it. But Steve, you mentioned when we were talking offline that even though this is a new finding, it sort of fits into a long lineage of this type of research, right?
Yeah. I mean, we could say we've been here before, right? And just for background, I mean, I did a little bit of ALS research early on in my neuromuscular career, and I've been in meetings with pretty much all like the ALS researchers in the Northeast, of the U.S. So I get a pretty good insight into the thinking and what's going on. Just say very quickly, many, many times we find stuff that is happening when the motor neurons are dying that potentially could be a clue to what's driving the disease, and yet the treatments don't have any effect. Treatments based upon that. Researchers are very cautious. And while this may seem like a slam dunk or it may seem very promising, this is not any more promising than a dozen other things that we discovered about ALS that did not turn out to be like the answer.
Right.
You also mentioned that based upon this data, it appears that this mechanism is quote unquote driving the disease, but we have to be careful about that because you could think about causes in different ways. Something might trigger or instigate a degenerative disease process or a progressive disease process. It might drive that disease process, meaning without it, it would not progress, it would not continue, or it may exacerbate that disease process. I think actually my reading of this, that this is more likely to be an exacerbating factor than a driver.
So this idea of like sort of a diathesis stress, that there's something genetic that tells the disease it's going to happen, and then this makes it either happen quickly or it gives the patient longer.
Well, only about 15% of ALS is familial, right? So we're talking about sporadic ALS in this paper, which means
but even
not
sporadic
genetic.
ALS, does it really mean not genetic, or does it just mean not heritable?
Not heritable. It doesn't mean there's no genetic predisposition. At most, there's a predisposition, or some people might react differently. But it's not a genetic disease, right? It seems to be sporadic, like there is no predictive family history at all. Just strikes at random. So sporadic ALS isn't one disease. It's just describing a syndrome. If your upper and lower motor neurons are dying progressively, you have ALS, whatever's causing it. So there's definitely multiple diseases thrown in here. So that's important to note as well.
Yeah. And it's probably hard to know if like autoimmunity is so complex. Is it, like you mentioned, that something happens in the body that sort of switches on an intense autoimmune cascade, or is something else happening?
The disease could trigger the immune system, which then you have a secondary autoimmune response in some people, which then exacerbates the original neurodegeneration. May predict, again, their survival, how quickly it progresses, but it doesn't mean that they will be fine without it,
Yeah, and that's the interesting thing, I think, from this study. Yes, okay, this idea of the CD4 T cells attacking this protein, that is really interesting, but that this subset of patients who had a robust protective response actually correlated very highly with patients who had a longer survival time.
Yeah, that's interesting. But
to me, that's super interesting. Yeah.
At the same time, again, this idea that ALS or a subset of ALS might be autoimmune is decades old. Like, this is not a new idea.
Yeah.
And even 30 years ago, I remember this research ongoing. For example, we've studied intrathecal IVIG. You use IVIG as a broad spectrum, powerful anti-inflammatory treatment, and you put it in the spinal fluid. that's surrounding the brain and the spinal cord, and zero effect on ALS. That doesn't mean it's not autoimmune because there's so many components of the immune system. But we can go down the line, and even with the newer, like really powerful anti-inflammatory, anti-rejection drugs, we've tried them. We basically try all of them in ALS, and so far none of them have worked.
Yeah.
Again, doesn't mean that this may give us a very specific mechanism that may be the only one that works and all other immune modulating therapies would not work. I'm not enough of a neuroimmunology specialist to know if that's the case or not. But the fact that a long line of anti-inflammatory treatments, including T-cell based treatments, have not worked is a huge note of caution here.
For sure. I think the hope, the cautionary hope, which we see all the time as research churns forward and is built on the backs of other research labs, is that it's all about getting down to being more and more specific. It's identifying targets that maybe weren't looked at before due to outcomes of other research. And so what they're hoping for in this finding is that these specific inflammatory CD4 plus T cells, which are specifically binding to the C9orf72 proteins, and these very specific anti-inflammatory CD4 plus T cells collectively may offer a new therapeutic target. And they also mentioned that, in this field of neuroimmunology, this is a big field, right? And there are implications for autoimmune components, or at least we could say immune responses in a lot of neurodegenerative diseases.
There's always going to be inflammation. And it's never going to be a good thing. I mean, completely, right? But is it, again, driving the disease or just maybe exacerbating it versus whatever? Yeah, I think it's complicated. It's a lot of complicated stuff happening, and none of it is the cause, right?
Absolutely. And I think when it comes to the world of therapeutics, it's important to remember that even if we're not able to get to a root cause, even if these are, like you mentioned, syndromes that are much more complicated than like a Huntington's, where we can go in and genetically modify the genes that cause it.
Which we did recently, by the way.
Yeah, I know, which is amazing.
We kind of missed that news item in the cycle, but we basically cured Huntington's with
CRISPR. Yeah. It's amazing.
Wait, can we pause there real quick? When you say cured, like what is the actual thing that's happening?
Huntington's is a very simple genetic disease. Like, we know exactly what causes it.
It's a trinucleotide repeat. Basically, it's 3 base pairs that repeat over and over again. And in each generation, the number of those repeats increases. And the more they increase, the more rapidly the disease progresses, the younger it presents and the worse it is.
Oh, that's interesting.
Right? So
it's a dominant disease.
It's dominant, yeah. So what they basically did was used a genetic modification to get rid of most of those repeats. And they essentially, in the patients they studied, they slowed it down to 25%. So if normally you would survive for 20 years with the disease, now you would survive for 80 years, which is not a cure, but it's pretty damn close.
Right. There's a ceiling for all of us at one point. So, that's fascinating.
And especially since we can easily check for it, genetically test for it, we can identify if you have the trinucleotide repeat when you're young, give you this treatment. And here's the other thing. It appears to be a once-in-your-life treatment, because once you do it, the neurons don't turn over, right? You keep your neurons for life, basically. So they just have to do it once. It's a surgery.
I wonder if you did it more than once, would it do another 25%?
Yeah,
does it have returns?
But it's it's an invasive, brain surgery to do it.
Yeah.
So, but what they showed was, apparently you only need to do it once. It slows it down by 25%, and this is a total game changer for Huntington's.
Oh, completely. And the thing to remember, I think, about Huntington's, just to remind folks, is that it's really rare to pass on deadly diseases that are dominant, because most deadly diseases that are dominant obviously kill the person who gets them, so they can't reproduce. Huntington's is weird in that it has a delayed presentation, so some people don't know until after they've produced offspring until after they've given birth. And then it's like, oh, crap. So that's why it's such a devastating disease for a lot of people, because there's a lot of psychological guilt and shame and just really complex emotions involved. So knowing that there's a therapeutic that, basically is a cure. And that's why that's different from things like Alzheimer's, Parkinson's, ALS, where these are also neurodegenerative diseases, but they appear to be more syndrome-esque. There's a lot of different things going on. But let's say development of a therapeutic that actually counters the intense immune response does occur based on this research. Fingers crossed, right? Best case, even if that's not a cure, it could help people live a Stephen Hawking lifetime versus...
That would be amazing. We got excited when we slowed ALS down by 20% and people lived for 3 more months. Statistically, that was a disease-modifying treatment. And if this could slow it down by years, that's huge for patients with ALS.
Yeah.
Hey, Lee, how you doing over there?
I'm doing great.
Learning a lot?
Go ahead, Lee, continue.
Yeah, ALS is in the news a lot the last couple of weeks because, Eric Dane got diagnosed.
Mm-hmm. Really?
Yeah.
He was
McSteamy
on Grey's Anatomy.
Yeah.
Okay. Yeah.
How old is he? Because most people are diagnosed after 50, right? But there are younger.
Yeah, I think he's like 53. He was born in 72.
Mm-hmm. Oof.
Man.
It's tough. Yeah, it's tough.
It's a terrible disease.
Mm-hmm.
All right, Bob, tell us about the complex chemistry they're finding on Enceladus.
Yeah, this was a fun topic to research. All right, so a re-examination of Cassini data from Saturn's moon Enceladus reveals even more reason for optimism for the potential of life on that moon. This was recently published in Nature Astronomy by a multi-institution team. The name of the paper was Detection of Organic Compounds in Freshly Ejected Ice Grains from Enceladus's Ocean. All right, quick backstory. Of course, I'm lying. This is not going to be quick. The story goes back as far as 2005. I actually found an old article that I probably read back then. And it sounded like the name of this article sounded like the name of a sci-fi series. It was The Fountains of Enceladus, right? Doesn't that sound like a title of a science fiction book? It describes the now famous Cassini mission, its epic discovery of ice particles erupting geyser-like in these jets on Saturn's moon Enceladus. The jets of water were bursting from cracks in the ice, and the resulting ice grains would be, shot up, into space. Eventually, many of them fell back onto the moon, or they could have also left permanently, becoming part of the E-ring of material that traced out Enceladus's orbit, actually. Because if you looked in the middle of the E-ring, like, oh, there's Enceladus. Now, if that wasn't cool enough, the obvious implication here is what? Where is this water? Where is this coming from? There could be a hidden ocean beneath the ice supplying the water for something I found out today. They found 101 geysers ultimately. So this isn't just one geyser, 101 of these. Now, that fainty, icy E-ring, you've heard about the different types of rings around Saturn. This E-ring has been known about for decades. And now, back then we could say that this ring was almost certainly coming from Enceladus and coming from these geysers. That's what it was proved way back in, or shown very strongly way back in 2005. So over many years of the Cassini's mission, the data from the Enceladus, the geysers, and the E-ring information just got increasingly compelling, especially regarding the potential for complex chemistry happening deep within the moon in that ocean. So here's some of the highlights during Cassini's tenure in the outer solar system in 2009, or at least for Enceladus specific data. In 2009, it showed that this deep ocean almost certainly had salt water, very important. In 2015, there was evidence announced for a deep hydrothermal activity. That's where hot rock and seawater interact. We've got these hydrothermal vents on the Earth. So when the hot rock and seawater interact, good stuff can happen. In 2017, chemical energy was announced in the form of a molecular hydrogen that was detected in the plumes. This is what hydrothermal vents create. And most importantly, this is a potential energy source for microbes. At least on Earth it is. So it's like finding whoppers coming out of this place on the Earth. Like, yeah, that's a good energy source for whatever might want to live on that stuff. And then let's see, in 2017, after Cassini was scuttled right in the clouds of Saturn, you remember that one? Like, okay, we're taking a dive into Saturn, and Cassini's never coming back. Scientists continued, even after the death of Cassini, they continued to mine and analyze this huge treasure trove of data and to create new discoveries. Like, for example, they found that large organic molecules was hidden in the data. They showed, this showed very rich ocean chemistry. It would not life, but life friendly for sure. Life adjacent, maybe? I don't know, but very interesting stuff. And then in 2023, Steve, phosphorus, that was a big find. Phosphorus was found in higher concentrations than we find it in Earth's oceans. And it's absolutely key for Earth life, or life as we know it on the Earth. That was a very nice find. So, okay, so this is where this latest research comes in. And for these researchers, this involved a reanalysis of the young ice grain data opposed to the much more common analysis the researchers did back in the day of these old ice grains that were orbiting in the E-ring. The problem there was that these E-ring ice grains could be orbiting for years or even centuries. And when you're in the space that long, what happens to things? There's space weathering. There's actually a thing called space weathering, and that could alter the molecules in weird, not understood ways. So the researchers wanted to remove that potential complication of the space weathering and re-examine the data on fresh grains that may already be existing somewhere in the data. And ideally, they would want to find these ice grains that were just recently shot straight from that ocean, from the subsurface ocean, and detected almost immediately by Cassini. And that's exactly what they did. In 2008, Cassini did a very rare, a very fast flyby of Enceladus right through the plume of particles. The scientists thought that the new models and their new filters, and just the raw Cassini data experience that these scientists have. They've gone through many years of looking at the Cassini data, examining it, studying it, testing models. So all of that experience they could now bring to bear on this new approach to looking at these fresh ice grains coming out of those geysers. And they were hoping that they could tease out, obviously, more details about what kind of chemistry could be happening in that mysterious ocean. And that's what happened. They focused on one specific orbit Cassini made through the plume. The orbiter was moving at about 18 kilometers a second, so it was just faster than I think it had ever done around Enceladus. And it was only 25 kilometers above the surface, 16 miles. I mean, that thing was just skimming right over the planet, essentially, super low. So it examined, ultimately they found that they were examining, or Cassini was examining ice grains that were only minutes previously were in the subsurface ocean. So within minutes, they came up from the subsurface ocean and were ejected out. And then there was Cassini just 16 miles away, ready to detect them and figure out, what it was composed of. Obviously there's ice, but what kind of hidden little gems might be in there that we hadn't seen before. So these were the youngest and the freshest ice grains ever detected. That high speed was actually an advantage because typically Cassini wasn't traveling that fast. So when these ice grains hit the detector at like 18 kilometers a second, essentially, right? What it did was it removed this kind of weird water fog that typically obscured the data, right? Because you had a lot of these water molecules grouped together, and it kind of created a fog, just an obscuring fog that could potentially mask some interesting data. So it obliterated those water molecule collections, and it kind of revealed essentially whole new organic families that just popped out. That these new analytical tools that they could see for the first time. So, but I mean, we're talking about organics like, you may have heard of some of these esters, ethyl groups. Most intriguing was the possibility of mixed nitrogen and oxygen bearing compounds. That sounds like very interesting detection there. It's not as much of a slam dunk as the other ones I mentioned, like the esters and ethyl groups, but it seemed fairly solid. What they found though was no smoking gun for life. Let me make that clear. They did not find any big hints of life at all. What they found were ingredients, not organisms, but these ingredients were very compelling. This wasn't just simple chemistry that was happening there. Definitely a few steps up above beyond that. So ultimately what they did was to strengthen that link between the organic molecules directly to that deep ocean, right? Because remember, these are the freshest ice grains that were being ejected from that geyser. So they had no time for any space weathering to change the molecules in some weirdly unpredictable ways. So these ice grains were directly from that ocean. And what they found is more families of organic molecules, better, just clear. Everything was a lot clearer and fascinating. And it just strengthened that link that there is something going on. For me, this is more exciting to me than those minerals that we recently talked about that they found on Mars. This is a lot more interesting to me. Here we have really good evidence of some kind of complex chemistry creating organic compounds right now. This is happening right now as we talk, as we record, as you listen to this, happening right now in our solar system. And this is not on Earth. In light of this data, which is, I admit, this is more of an incremental change, could you guys think of a more promising location for life in our solar system right now other than Earth? I mean, there's Enceladus,
Europa?
there's Europa.
Europa is the only other one, really.
Yeah, Europa and Enceladus are the big boys. I would put Enceladus over Europa, but there's also Titan and Ganymede and Callisto. They're not as strong as Europa and Enceladus, but they're also, intriguing, and there's some solid hints that something could be going on. I would move Enceladus to the top position right now for sure, even over Europa. So my big hope here is that this is going to strengthen the case for the European Space Association to mount a mission to orbit and land on Enceladus, because I didn't take too much of a deep dive on what they're actually doing about mounting a mission, but I think they've at least been talking about it and been planning it at high levels. But I think this could be the data they need to we really got to go over there, man. There's something really cool happening that we need to get close to. And the reason why they would want to get there is because when we go back there, they could bring real chemical toolboxes to bear to examine the compounds before, this is important, they could examine them before they're smashed apart, like our earlier techniques did. So I'm really hoping that we can get back there before I'm dead and see what the hell's going on in Enceladus. So
you're
cross your fingers.
Saying get your ass to Enceladus.
Nice.
Yeah,
nice
so to
one.
recap, it's got liquid water, a source of energy, it's got complex organic molecules, and it has the other chemical elements that are useful for life. It's all there.
It really is.
It's almost, we would have to figure out why life isn't there if we don't find
Yeah,
life
right? Because that
in
would
the
be, I'd be shocked. I just want to know. I mean, this is why I wish we had superheroes. Because I'd be like, Superman, go to Enceladus right now, pick us up a sample and bring it right back later today. And that'd be great. Because you know what it takes to get there. Millions of dollars. The stupid chemical rockets are going to take years.
Yeah, but it's worth it.
It's worth it. It's worth it. But, hey, man, when you're
just
62, you want shit to happen faster.
All right, Lee, you're going to tell us about the Genius Act.
First off, maybe I talk a little bit about what I do and then talk about the Genius Act. I'm in venture capital. Venture capital invests in really early stage businesses. It's considered an alternative investment class. Basically, how do companies, when they first start with a pitch deck or an idea, how do they get capital to expand and grow to get revenue? Once they have revenue, how do they expand and grow into the market? In July of 2025, the Genius Act was passed. It is going to take effect in January of 2027, and it allows people who aren't necessarily the richest people in the world. It allows the managers of their retirement plans or their pension plans to divert a certain amount of capital into earlier stage businesses. And so, if you have a 401k, you have an IRA, a lot of listeners wouldn't classify to be able to, invest in venture itself, but now you'll be able to.
So reading about it seems like the part of this has to do with stable coins and cryptocurrency. What role is that playing?
Yeah, so crypto and stable coins is a big part of it. But I'd say, there's going to be a lot of people in the venture community that are going to try to get access to that large chunk of capital. Venture spending has been primarily dominated into AI over the past 3 years. And so there's a lot of, businesses that are looking for that capital, a lot of investors that are trying to deploy capital into that part of the asset class. And so many large VC firms are becoming registered investment advisors currently. People are trying to figure out, how do I tie my VC fund into RIAs that can allocate a certain percent to my to venture capital. And so, I'm getting into the intersection of sort of investing and skepticism and critical thought for the first time in my life, which I know that sounds silly, but cool. There's a lot of potential risk around some of the things that we're investing in, from a technical feasibility, from a scientific feasibility. And I think people should be concerned about it.
And so should we be concerned about this act? Like, what do you think is going to be the downstream effect?
Well, I think they're going to put a lot of restrictions on the amount of capital that can be diverted from a portfolio. So probably going to be 1% to 2%. The asset class as a whole performs okay. It's very illiquid. You cannot typically get an exit or get money back for a 10 to 15 year period, which may not be appropriate for some individuals' retirement horizon. So they're going to limit it a little bit. But I would say it looks like a gold rush for venture capital. And there's a lot of people trying to get access to that and really changing their business model in a way that allows them to bring down new capital. And I'd say, you mostly think about venture capital as a bunch of billionaires putting money into these high risky, early stage startups. And it's not necessarily true. Venture capitalists don't use a lot of their own money to run these large funds. And a lot of those large funds, already get a ton of capital from, pension fund managers and things like that. This is a whole new way to get access to 401ks and IRAs. And then the type of investments we're seeing a ton of investments in things that may have a 20 to 30 year horizon. A lot of there's been a huge bubble in AI. A lot of us are skeptical. We think that AI is going to be great. Do we think it's going to be as great as it is, presented? Probably not. And so there's a lot of hype around it. There's a lot of hype, with quantum. Do we really know where we are in quantum? I have my own questions. There's certain research things that has to happen. There's certain algorithmic development things that have to happen. And if you're looking at a 5 to 10 year horizon, which was traditional venture, back in the 90s and early 2000s, it's really questionable whether, those types of investments will get a return, because in the private markets, when you do an investment, that money is basically given to the company. And it's very hard to sell those positions. There's often no buyers until the company either goes IPO or some large other company acquires that business. The investors don't get their money back.
Yeah, I mean, obviously, I'm no expert. I've actually talked a lot to another skeptical investor, Phil Ferguson, about these very topics. And all my money that I have control over is in index funds because I'm scared. And so for me, I don't know, these kinds of investing a lot in crypto, I don't know, it scares the crap out of me. And maybe I sound like I'm older than I am, but I'm at a stage in my life where I just can't afford to not get returns on, my retirement.
Yeah, and I think, fully diversified, I mean, some of the best advice I ever got was, take $200 a month when you're in your early 20s, put it in an index fund, and just do that until you're 45 or 50, and you'll be a multimillionaire. And I think that that's good advice. And there is some advice that most of the gains in these companies are going to be realized before they ever hit the public markets, right? And so if you're in an index fund, you're in something that's public most of the time. And so most of those gains will be given when it's a private company. And so you could use the same logic. But I think what we see in the industry is a lot of, to get a fully diversified portfolio in venture, you have to be in, 1,000 or 2,000 deals, maybe 500. What we see in venture is sort of these one-off, I'm going to gamble here and get 1,000 to 1 or 100 to 1. And it's a real long shot. But that's also what, brings the capital into the markets.
Right. And so the people who can afford that kind of risk are the ones who are taking it.
That's correct. And before this, I would say that, there was a lot of pension fund money in venture. And it's, it's a 1%, 2% allocation, but those funds are huge. They typically focus later in sort of the growth stage of the business. But now they can focus at the earliest stages where you can have just a team with a pitch deck. Maybe they've got a product built.
So what do you think about one of the main criticism that I've read is that this is really just a way for tech giants to make money off of crypto with less regulation?
Well, I've made more money in my life than anything with crypto. So I have a little bit of bias there. But it was more just about being interested in the technology and sort of, buying and holding over a long period. But I do think that's right. Tech giants, it's almost a shell game. Let's say that you have an investor, you create a large tech company, you fill it with the board of other investors, and those investors will invest in a new company, and then they sort of advise that company to buy that startup. So they get these amazing returns, because they have that connection. And so the best performing funds have that sort of baked in success that they've created for themselves through, years and years of that shell game. But, I agree. A lot of the projects, not vast majority of projects in crypto, you often don't know who is running them. I have a crypto project that's a sports betting project, and the people don't show their face on podcasts, or they won't give their name because there's that anonymous nature to crypto. It just doesn't seem like for the stable coins it's a little bit different, but I'm pretty skeptical if this is a way to get into sort of this Ponzi scheme of crypto investing. That should be terrifying for everybody.
Yeah. And for the audience, stable coins are crypto that's tied to a currency. Is that essentially correct?
It can be tied to a currency. It can also be algorithmically tied.
Okay.
So what makes it stable? Or is that a misnomer?
Well, when it's tied to a currency, when they're actually, purchasing fiat and sort of balancing the equation so that it's backed by fiat, then it's a good stable coin. You might see others where it's tied to some other asset class. But they're, a few years ago, they were just algorithmic. And I didn't understand all the details, but basically they could or could not purchase a certain amount, or they had the ability to purchase certain amounts. And a lot of those crashed. I was actually in Puerto Rico pitching a hedge fund at the time, a crypto hedge fund, which would have done very well. But it was during one of the last crashes, and it was because some of these algorithmic stable coins took a dive to zero.
Not so stable.
No.
All right. Thanks, Lee. Jay, it's Who's That Noisy time.
All right, guys. Last week I played This Noisy. What do you think?
The only thing I could say is that there appears to be a Doppler effect in there.
It feels like something launching.
Those are both good comments. Bob, Lee?
Sounded like a jet to me.
Well, a lot of people guessed some type of aircraft. So a listener named Dan Lee said this week's Noisy is a conveyor belt loading luggage onto an airplane as another plane takes off in the background. Dan, you are incorrect, but I like where you went with that. And again, it's a very good guess that it's an airplane sound, but his guess was incorrect. Matthew Morrison wrote in and said, "Hi, Jay. My daughter Nev thinks this sounds like an airplane flying over a rainforest." And again, good guess. You're not in the exact right place, but that is definitely a good guess. We have a listener named Bob. Bob! I believe this is the sound of a jet fighter taking off from the deck of an aircraft carrier. He goes on to describe, at the beginning, you can hear what sounds like a cable slapping around on some metal. I believe that is the catapult used to get the jet to the proper speed for takeoff. Also incorrect. I have another listener named Bob. Yeah, two Bobs. Bob. This is Bob Marshall. He said the sound is recorded in part in the Welsh Valley called Mock Loop, and this is where you can sit on a peaceful mountain and watch a wide variety of military jets pass below you in the valley. Because apparently they do a lot of practice flying and everything. So that would be very cool. Sorry, Bob, also not correct. Another listener named Alex Freshy said, "Hi, Jay. I'm going to guess this week's Noisy is a hypersonic wind tunnel. The cycle time for fast wind tunnels is surprisingly quick. Thanks for all you do." Yeah, check that out, guys. A hypersonic wind tunnel. I had never thought of that before. What the hell? That must be...
I thought they were impractical. Can they get them hypersonic?
Just because he said it doesn't mean that they necessarily exist, but it is a cool guess. But that is incorrect as well. So what are we dealing with here? So in the beginning, you hear what I would say is kind of like a little rumbling kind of sound, and then it builds into like a very fast, what Steve described as a Doppler effect noise. In the end, guys, this is a roller coaster.
Oh.
So this one is called Falcon's Flight, and it happens to be a record-breaking roller coaster that's being built at Six Flags in Qiddiya, Saudi Arabia.
Oh, wow.
It's designed to be the tallest, fastest, and longest coaster in the world when it opens. So it gets up to 155 miles per hour.
Oh,
that's 250 kilometers an hour, which makes it the fastest one on Earth.
That's terrifying.
It has a 640-foot drop, or 195-meter drop. That's higher than any existing coaster. It is nearly 2.5 miles, or 4 kilometers of track, which makes it the longest roller coaster. And the unique feature here is they say the ride will plunge off the side of a cliff using the natural terrain to create extreme height and speed.
Oh my God.
Which is very cool. It's going to be opening when that Six Flag apparently opens, and it is currently under development. So let me play that for you again, and just give you a visualization. So imagine the roller coaster is running on the track, and it comes over the lip of the cliff and then goes straight down. And then it picks up all that speed, and then it goes up a huge ramp, and that's when you hear that Doppler effect.
How many people are going to pass out on this thing?
Yeah.
I wonder how many G's you push.
Yeah, how many G's are you pulling?
Yeah.
And now that you know what it is, you can imagine what's happening when you hear this sound. That's scary.
And it makes me think of something, right? So all my life, I thought that what I was largely hearing from a jet was the engine. And this sounds like a jet, but it's not an engine. It's, okay, there's wheels of some sort on a track of some sort, right? But, it also might just be the wind breaking off of the front of the coaster.
Wow.
It makes a jet sound. That is, pretty damn serious. I don't know. Bob, would you ride it?
I don't think so. And I can't imagine. That's a little too. I'll go on almost any ride. That seems a little extreme. I'd have to see it, and then probably.
What if
really
I
not
double
go.
dog dared you? Would you do it?
Got to do it now. Jay, for what it's worth, I confirmed that hypersonic wind tunnels do in fact exist.
Very cool. Thanks, Bob. Hey, Lee, have you ever been drunk on a roller coaster?
Never.
Good man. Yeah, you don't want to do that.
I was less than 21 years old when I was on the roller coaster.
Well, you don't want to particularly be drunk on a roller coaster that sounds like a friggin' jet. That's my advice to the world. I have a new noisy this week. This comes in from a listener named Paolo Ciaroca. Yeah, Ciaroca. Thanks, Paolo. And here's the noisy. Well. So if you think you know what this week's noisy is, or you heard something cool, you can email me at wtn@theskepticsguide.org. Steve, as we speak, I'm planning 3 more SGU Private Show Pluses and extravaganza weekends that we just did in Kansas. 3 more.
L.A., Sydney, and Christchurch.
No.
Oh.
No. We are talking about Seattle. Mm-hmm. We're talking about somewhere near Milwaukee in that region. And then we are talking about New Haven, Connecticut.
Oh, those 3.
Yeah, I'm not even getting to next summer.
Yeah, I was talking about next summer.
Yeah, there's going to be a lot more activity going on then. That is going to be the 2 conferences that we're going to be attending. First one is in Sydney. That's going to be on July 23rd and 24th and 25th. And then we're going to be attending another one, which is the following weekend. That'll be in Christchurch, New Zealand. And there'll be lots of events, secondary events surrounding that. I'm going to give out all the information coming soon. We are finalizing. Everything takes longer than you want, but we're there. I just got to do the final work to turn on the ticket sales. It's going to happen soon. 2 more quick things. If you appreciate the work that we do, you could consider becoming a patron of the SGU. You can go to patreon.com/Skeptics Guide and join us on our adventure to educate the world and get rid of every single thing that irritates all of us.
And you know what we haven't mentioned in a while is if you want to learn more about skepticism, you can buy both of our books, The Skeptic's Guide to the Universe and The Skeptic's Guide to the Future. The Skeptics Guide to the Universe is basically a primer on scientific skepticism. Quite a fun read. And then in the Skeptics Guide to the Future, we apply all of that to thinking about future technology. Very interesting. So, those books are still highly relevant. You should not only get one, but buy one for everyone you know.
Mm-hmm. Got it.
I have people asking me on all different platforms, what is going on with the new podcast? So let me give you all the base information.
There's a new podcast?
Yes. It is called the Political Reality Podcast, and this podcast will be hosted by Steve, and it'll be hosted by Andrea Jones-Roy. Steve, what the hell is this podcast?
So Andrew and I we are deep in the weeds of scheduling out at least the first season of this podcast. We're going to be talking about a lot of political issues, but from a very, academic and neutral point of view. Doesn't mean that we're not going to, call it like we see it, but it's more about like the meta discussion of politics. Andrea said, this is the civics class that everybody should have taken but didn't.
Nice.
But try to make it as timely and relevant and in the news as possible. She has a ton of great guests lined up, all experts in political science and different areas of politics. So basically this is, if you want to have an informed opinion about something, we don't care what your opinion is, but this will help you have an informed opinion about any political issue.
Nice.
All right. Well, thank you, Jay. We have a quick email. This one comes from Gary. And Gary writes, parrot as intelligent as a chimp. And then he links to some experiments done with a Kia. Do you guys remember the Kia, K-E-A?
No, what is that?
Yeah,
That's
that
a
sounds
bird.
familiar.
Is that a bird in New Zealand?
Yeah, Gary's from New Zealand. It's a giant parrot of New Zealand.
I remember that now.
Remember seeing him on the side of the road? We're driving around in New Zealand.
No, that was when we opened up the van door and he came running over to the van door, demanding attention and food.
So you guys have to watch this video. And it's a series of experiments where they're trying to determine the problem-solving and predictive abilities of Kia. So the basic setup is there's jars that have either red or black clothesline clips. Like you take half of the clip, you know what I mean, without the spring, that little piece of wood, and half of them are black and half of them are red. And then you have one person who picks from one jar, another person who picks from the other jar. And they're trying to see if the parrot can predict which person is more likely to have the black piece of wood. Because they pick one hand, they get the wood, and then they can exchange it for a treat, but they can only exchange the black ones for treats. So you get the setup? Mm-hmm. So the parrots want the black ones. So they do all kinds of permutations, including obviously picking it out of a jar with mostly black ones, picking it, the other person picks it out of a jar with mostly red ones. The parrot knows it's more likely to get a black one from the jar with mostly black ones. That's easy. But then they keep going, right? They put in a barrier. So even if the bottom half of the jar is all black, the top half isn't black, the parrot won't fall for it, right? So it's not just going for the color. It knows that it's only picking from the top. You get that? And then they're on some cases, the person looks at the color of the stick that they're picking, and the parrot could learn which of the people doing the experiment prefer the black pieces of wood. So they can actually... So in other words, if it's me and you, Kara, and you, Kara, you look and always pick a black one, and I don't look and it's random, the parrot learns that you always have the black one. And so then it will pick you even if I'm picking from a jar with more black sticks.
Right. Yeah, because it's good at statistics.
Yeah, so right. So, but here's the upshot of it is they do as well in this experimental paradigm as chimpanzees do.
Well, and that's the thing about these headlines that are like so-and-so as smart as so-and-so. It's like, how are you defining intelligence?
In this very specific paradigm.
Yeah, they're good at this. They're better at this task than a chimpanzee, which there are all sorts of animals that are better at specific tasks than human beings. You know what I mean? It doesn't necessarily mean they're more intelligent, or maybe they are in that one definition.
Yeah, I was wondering if I could borrow some of these parrots for venture capital.
Yeah.
Seriously.
There was that study where a chimpanzee, a monkey, basically chose stocks and did as well as most of the professional stock pickers. But maybe we could try it with the parrot. No, but the important thing here, Kara, is that birds rule. That's the important thing.
Birds are really smart.
Let's not lose sight of that. All right. Let's go on with science or fiction.
It's time for science or fiction.
Each week I come up with three science news items or facts, 2 real and 1 fake, and then I challenge my panel of skeptics to tell me which one is the fake. There is a theme this week. The theme is evolution. You guys ready?
Let's do it.
All right. And you'll see that there's a sub-theme within evolution that'll become apparent very quickly. Okay, item number 1. Leglessness has evolved independently in lizards at least 25 times. Item number 2. Although the precise number is debated, camera-type eyes has evolved from more primitive eyes independently in vertebrates about 4 times. And item number 3. Complex multicellular life has evolved independently on Earth at least 5 times. Okay, Lee, as our guest, you get to go first.
Leglessness has evolved independently in lizards at least 25 times.
Now, I will clarify one small thing there. Leglessness doesn't necessarily mean there's 0 leg left, because even snakes have leg bones, and even some snakes have primitive little legs sticking out. It means what legs they have are vestigial. They're not using them as legs.
Yeah, I guess like that could be true. I mean, we all know lizards hate pants, so independently, I think that could be true. Although the precise number is debated, camera-type eyes have evolved from more primitive eyes independently in vertebrates about 4 times. Yeah, sure, why not? I think possibly, yeah. The one that really is setting off my antenna here. Complex multicellular life has evolved independently on Earth at least 5 times. I just don't know how we get the evidence for that. So I think that, to me, is the fake one.
Okay, Jay.
All right, well, I might have some questions to you. Well, the leglessness has evolved independently in lizards at least 25 times. I mean, that's interesting because, we're talking about millions and millions and millions of years. And would that develop independently over all that time? Like, I guess it would be like when you say independently, like it started off in one species and then another one developed it, and they didn't co-evolve the whole thing.
Yeah, there's no common ancestor with leglessness. It happened completely evolutionarily independently.
And why is this, when you say leglessness, meaning they don't have legs or they drop their legs?
They don't fall off. They have vestigial or no legs, basically. Okay,
roger that. With my understanding of nature and evolution, I think that this is plausible because I am aware that similar evolutionary traits have happened in other species that don't have a relation, right? So why couldn't it apply to this? So that one is tentatively science. The second one is weird. I got to ask some questions. Although the precise number is debated, camera-type eyes have evolved from more primitive eyes independently in vertebrates about 4 times. So when you say camera-type eyes, what are we talking about?
So not compound eyes like insects.
Like our eyes.
Yeah, like our human eyes.
Yeah.
Yeah,
right.
What? Lens, retina?
Yeah, the basic setup of pupil, lens, retina. That basic setup.
That would be really cool. Now, here's why I'm not sure about this one. Because if that happened, and with all the debates that I've heard, people talking about evolution and, about the fact that, what's it called, Steve, irreducible complexity?
Mm-hmm.
Why wouldn't someone on the evolutionary side, or why wouldn't I have heard someone bring this up? Hey, man, it's happened 4 times independently from each other. Because that would be a great argument. That doesn't mean it didn't happen. It just means I have not heard that. And that would be something I would think that I would have heard at this point. So that's more of a fiction than a science for me. And then complex multicellular life has evolved independently on Earth at least 5 times. That one also, you think, all right, doesn't everything kind of all go back to the same thing? But I'm not so sure about that. All right, I'm going to go with the camera type as the fiction, Steve.
Okay, Bob?
The lizard's leglessness. Yeah, I mean, I've seen images of lizards with clearly like weird, like, oh, they got front legs but no back legs, or they're clearly vestigial. The only question here for me is 25 times seems a little bit big, but I could totally go with that, especially considering the other two here. The second one here, the camera eye. And again, absolutely, in my mind, there's no question about this, but 4 times might seem a little bit low. Then you are focusing it on vertebrates. So that narrows that down a little bit. But sure, I could see that. And then the third one, see, this is the one that got me. I think I agree with Lee, right? You picked this complex multicellular life as the fiction. This is the one that grabs me. I mean, it took how long? I mean, single-celled microbial life went for so long, millions and millions of years, even what, was it a couple of billion years potentially? And then all of a sudden multicellular life appeared. It took a long time, really long, depressingly long time. I don't think we have any evidence that it evolved independently at all. So this is the one that viscerally grabbed me. Doesn't really make much sense. And even if we did know that it happened, what would the evidence be? I think all multicellular life has a common ancestor. That's fiction, I would say.
Okay, Kara.
I think that what is kind of getting me on this is if leglessness has evolved at least 25 times, if that is science, I think these other two numbers are ridiculously low, even for complex multicellular life. I do think that it never ceases to amaze me when I learn about certain lineages, how many times certain things evolved over and over, simply because the pressures continue to exist. So the mechanisms might change, but the pressures on those, genes to be selected for continue to exist. So if leglessness in lizards evolved at least 25 times is science, then I think I have to be careful about the wording here. Multicellular life has evolved independently at least 5 times, I think is also science. But you said that camera type eyes have evolved from more primitive eyes independently. You did say invertebrates, so that is an important caveat, but it just says about 4 times. I bet you it's also at least more times than that. I think it's probably a lot of times. And so I'm going to go with the camera type lenses. I don't know if it's the same reason Jay is going with it, but I think it's probably more than that.
All right. So let's start with the first one, since you guys all agree there. Leglessness has evolved independently in lizards at least 25 times. You all think this is science, and this one is science. Yep, this is science.
That's good.
Lizards apparently like to be legless. And so there's 2 basic scenarios they think in which lizards evolve leglessness. One is burrowing. They go underground. And the other is on the surface of the ground. You're basically crawling along the top of the ground. And that does tend to result in some differences, like how long the tail is versus the thorax. But in any case, there's a lot of legless lizards, the western glass lizard, the European legless lizard. There's a lot of legless skinks. And they don't just form leglessness, they also form very snake-like anatomy overall. They kind of adapt to the snake-like predatory style, not just the
some of them
ones.
you'd be like, yeah, that's a snake. Like, nope, that's not a snake.
Well, some of them have evolved as mimics. Like, that's their defense mechanism. Like, skinks look like snakes as a defense mechanism because they are relatively defenseless. But if a potential predator thinks that they're a snake, they'll leave them alone. So it's not that adaptation might be mimicry, not functionality, if that makes sense. But there's also a lot of functional convergent evolution among lizards. One piece of that is leglessness. Now, why would I say at least 25 times? In all of these items, this is the same reason why there's debate, whether it's at least or about or whatever. It's because there's always at least these 2 points of contention. One is what's independent? Like, how independent does independent have to be? And what do you count as the feature you're looking for. Like what counts as leglessness? What counts as independent? So, for example, like how legless does the common ancestor have to be before you say it's homologous versus analogous, right? Homologous meaning derived from a common ancestor. Analogous means you independently evolved it, but they look the same. You know what I mean? Like if it was like partly legless and then spawned multiple clades that were truly legless, was that really independent, or were they halfway there? Does that count or not count? So anyway, there's no sharp demarcation line here, and that's why there's this fuzziness. But the general consensus is there's at least 25 lineages where it clearly independently evolved leglessness. The number could be a lot higher depending on where you draw your line. So that one's science. I guess we'll go in order. Although the precise number is debated, camera-type eyes have evolved from more primitive eyes independently in vertebrates about 4 times. Jay and Kara, you think this one is the fiction. Bob and Lee, you think this one is science. And this one is the fiction.
Yeah,
Good
Jay.
job, Kara.
All right,
Jay.
Kara, how
Now,
we did it. I can't believe it.
Let me ask you guys, how many times do you think camera-type eyes have evolved in vertebrates?
A lot more.
8?
Jay?
1.
Or maybe it's like 25.
What'd you say, Jay?
1.
It's
said
1. The answer is 1.
Oh, so I was
one
okay.
time.
Only one time?
Yes, the common ancestor of vertebrates had eyes. That's all that takes.
Of course.
Yeah, so
Oh, so how
one
many times
time-
did it evolve in all life? Do we know?
give me a guess. So again, there's
have a
this
number?
what counts as an eye, what counts as
Right, right.
independent.
Okay,
But-
I'm going to go with similar to the legs then, like 25.
40 to 60 at least, and the number might be a lot higher. So if you're looking at all of life, there's at least 40 to 60 independent times where something like an eye evolved, right? So you have camera-type eyes in vertebrates and famously also in squid, right, as cephalopods. You have compound eyes in insects. You have lots of creatures have eye spots, etc. So like, does an eye spot count as an eye? So most people, biologists would say, yeah, if it's clearly detecting light, it's an eye. Even if it's a proto-eye or a primitive eye or whatever, it's still an eye. But again, you could nitpick about exactly where you want to draw the line there. Here's the other thing, though, about the independently part with eyes, which is very interesting, is that all creatures with eyes share some biochemical similarities. So it's possible that the common ancestor of like early on, like somewhere deep in the Cambrian or even the Ediacaran, somewhere deep in the Cambrian was a creature with the biochemical mechanism for photoreception. Mm-hmm. And then eyes evolved out of that many, many times, but those proto-photoreceptors were already there. The proteins were there. The genes were there. So is that truly independent? But they're counting that if the physical eye was independent, they count that as independent. But even though it might not be biochemically independent, does that make sense?
Mm-hmm.
Yeah. I could deal with that.
Yeah. All right. That's why I said this one. This one was so complicated I had to make it the fiction because I couldn't figure out a way to make it that would be not nitpickable. You know what I'm saying?
Yeah.
It was too many variables. So it was easy, that's an easy way to make something the fiction because I don't have to worry about it being true. All right. Item number 3, complex multicellular life has evolved independently on Earth at least 5 times. Bob and Lee, you thought this one was fiction, and this one, of course, is science. So what are the at least 5 groups of multicellular life that evolved independently?
They got to be the kingdoms, right?
So what are they?
Animal?
Animals,
Plant?
plants,
Vegetable, mineral.
Fungus?
Wait, how's that go?
fungus, and those are the easy ones. Now it gets hard.
Archaea?
No, those are single-celled.
Oh, wait, sorry. What were you asking specifically? Complex multicellular
life.
Oh, right. So it can't be archaea and it can't be bacteria.
Right. So animals, plants, fungi.
It's like blue-green algae, I think, is single
Protozoa.
Close.
Protists.
It's not protists or single-celled. Red algae
Oh, so it is a type of algae. Okay.
and brown algae. Those are the 5.
Freaking algae.
God damn.
There's a debate about a sixth, but 5 is, that's why I said at least 5.
Shit.
Yeah, and you have to
sense.
say complex multicellular life because there's a lot of simple multicellular life.
Yeah, like slime mold.
Yeah, exactly. So the number could greatly increase if you go down that complexity spectrum to include simple things.
My God.
Again, there's always demarcation lines in evolution. Evolution is messy, but this is generally accepted that these 5 things independently evolved complex multicellular life. Yeah, that item is what triggered this theme. I saw that. I'm like, I don't know that everyone's going to go for that. I'm going to put that in as one of the items, and then came up with the other 2.
Good choice, Lee. Good choice. God damn it, I'm pissed off, though. What the hell?
That brown algae. Ugh.
It always gets you.
That stuff.
All right. Well, Evan's not here, so I'm going to do the quote. I actually picked this quote even before I knew Evan couldn't be here.
Steve, do you mind if I do it?
Go ahead. Jace, do the quote for us.
The chimpanzee study taught us perhaps more than anything else to be a little humble, that we are indeed unique primates, we humans, but we're simply not as different from the rest of the animal kingdom as we used to think. Jane Goodall!
Oh, my God.
Yeah.
It's a blast from the past. That's a great... I searched through a bunch of her quotes. That's my favorite. I really like that quote because it is so true. Her work, Fosse's work, so many other researchers' work has showed us that we are animals, and every time we think there's something totally unique about us, some other animal takes us down a peg and shows that, nope, we're just a very interesting, but still an extension of the animal kingdom. Right?
Yep.
I agree with that wholeheartedly.
Good. All right. Hey, Lee, thanks for joining us. Did you have a good time?
Lee.
I had an absolute blast. This was great.
Yay.
And thank you all for joining me this week.
You got it,
Sure,
Steve.
man.
Thanks, Steve.
And until next week, this is your Skeptic's Guide to the Universe. Skeptic's Guide to the Universe is produced by SGU Productions, dedicated to promoting science and critical thinking. For more information, visit us at theskepticsguide.org. Send your questions to info@theskepticsguide.org. And if you would like to support the show and all the work that we do, go to patreon.com/skepticsguide and consider becoming a patron and becoming part of the SGU community. Our listeners and supporters are what make SGU possible.